Use calculated data when describing trend. S, F, D, P. Units. Draw cell walls as double line thicken of inner wall of guard cell. If mention partially permeable membrane, what moves in and what can't, mention diffusion. Errors. Subjective nature of deciding, qualitative method, similar concentrations look the same so can only be an estimate. Hard to judge color by eye, see when color starts to change, use white background color chart colorimeter. Difficulty in timing and sampling, observing all at the same time, so miss the color change, measure each separately. Difficult to determine when the milk had gone completely clear, mark 10 on underside, hold against dark background, comparison with endpoint, shine a light through. Difficult to measure height of precipitate accurately with a ruler, measure mass of precipitate, asterisk, have a white-black background or grid, use measuring cylinders or graduated test tubes. Overshoot of endpoint, add smaller quantities of iodine. No repeats, not enough trials, more samples, 3 or more trials should have been done and average results. Insufficient time intervals, XXX to compare with, increased number of intervals, range of comparisons. No control. Didn't measure. Drops could be of different sizes. Contents remain in test tube when adding to other tube, so volumes may vary. Tip and rotate not standardized. Same dropping pipette used, leading to contamination, effect, dilution of solutions, acid in wrong place, washing all apparatus as new dropping pipettes. Temperature didn't mention thermostaticula of water bath not maintained, insulate, use heat shield, add more ice. Drawing the line on beaker accurately, measuring height, measure volumes of water. Didn't stir. Dough was round, not flat, smooth, use balance measuring cylinder. Height of foam uneven. Bubbles of different sizes, measure volume of oxygen using syringe or measuring cylinder, mass lost. Some of the oxygen escaped before bung fitted tightly, foam reduces, use thistle funnel and tap to add hydrogen peroxide, tightly fitting bungs doing quickly closed system. Bubbles too fast to count, use a tally counter, method of collecting gas, measure volume, use 2 people, repeat for reliability. Dilution of peroxide due to washing, use new test tube each time. Potato sticks may have come from different source, use from same source. One potato stick was soaked in water and one was not. Cooked potato not cooked before adding to peroxide, temperature at start different. Cooked potato stick was not dried before adding to peroxide. Cutting the leaves cubes accurately, use a cutter of known size. Different distances from lamp, arrange equidistant. Chloroplast settle out, stir. DCPIP and chloroplast extract not mixed, stir or shake. Mixture of leaves, select leaves of same type. Contamination, damage to cubes from handling, wear gloves. Small cubes stick together so not all surface area exposed, separate with glass rod. Accuracy of calculating surface area of leaf, use grid with smaller squares. XXX measured at different times, stagger start of investigation. Difficult to measure distance between ends, use magnifier. Evaporation of salt solutions, cover petri dishes. Stems mixed up, stems labeled. Cutting to same length, use of ruler. Slice cut too thick, use of sharper cutting tool. Cut unevenly, cut halves equally. Pieces not submerged with liquid. Leave for longer time. Different levels of intensity of exercise will affect pulse rate differently, ensure exercise is of same level of intensity type. Longer period of exercise to see long-term pattern, tell whether it levels off or continues to rise. Results may be influenced by other variables, control temperature, time of day, Diet clothing. Error in counting pulse rate, use a pulse counter. Standardized time to measure pulse rate. Cardboard lids do not fit snugly on beaker, holes made in cardboard so heat loss. Start temperatures the same. Improve insulation of beaker. Measure volume of water in beakers. Squeezing regularly, consistent force of squeezing. Tape holes. Thermometer left in the water throughout the investigation as time taken for thermometer to reach the water temperature is longer. Same species woodlice. All animals healthy, not damaged. Control of one variable e.g. temperature, humidity. Methodology. Equilibrate temperature. Thermostaticula controlled water bath. Measure volume of DCPIP used pipette barrette drop counts. Shake after each addition of DCPIP. Make sure measurements of height of foam in each test tube can be compared. Measure from same point. Keep ruler straight. Measure at eye level to avoid parallax error. Rinse dialysis tubing in place in test tubes with distilled water. Methylene blue dye. Hydrogen carbonate indicator. Sodium hydrogen carbonate. Petroleum jelly. Remove tested germinate seeds to get extract of enzyme. Control temperature. Keep in the same room. Put into an environmental chamber. Control air currents, wind drafts. Keep all windows and doors closed. Control light intensity. Use a light source at a fixed distance. Control leaf surface area. Use leaves of same size, surface area. Heat shield. Check apparatus used to measure water uptake by plants does not leak. Control. Replace, with water, boiled enzyme, glass beads, dead maggots, not rotting, no maggots. Keep the rest of the experiment unchanged. Why drop of liquid in capillary tube? Use up oxygen. Soda lime absorbs CO2 released. Volume decrease. Drop in pressure, suction. Show water is lost. Method of collecting liquid, water vapor. Test for water. Anhydrous cobalt chloride paper test boiling, melting point. Result. Blue to pink, 100 C, 0 C. Why investigation repeated? Increase reliability. Identify anomalous results. May not be accurate the first time. Check method, technique. Why more than one sample used? Find an average. To see if measurements are comparable. To find anomalous results. How to obtain more accurate value for optimum pH temperature. Test at smaller intervals. Between X and X, optimum value from current investigation. Test at larger range. Why important to keep temperature constant? Rate of enzyme affected. Increased rate works faster aura. Temperature should be a CV, not IV. If temperature different, results less reliable. Why used large samples? Obtain a representative leaf size. Identify anomalous results. Why leaves selected at random? To avoid bias, gain a representative sample, so that a comparison can be made. How to use grid. Draw around the outline of the leaf on a grid. Place leaf under a transparent grid. Count the squares. Include any squares more than half covered. How to measure pulse rate. Position, e.g. neck, wrist, elbow. Artery. Close to surface, can be pressed against hard structure beneath or bone. Number of beats, pulses per unit time measured. How to collect a random sample and count the seeds accurately. Use same plant. Different locations, random numbering of samples, e.g. choosing numbers from table, pick fruits from bag. Collect seeds inside container or water, discard fruits that have already split. Tally chart, click counter, repeating count. Why measure dry mass? Water content in seeds is variable. For comparisons to be valid. Safety precautions, how to safely carry out a test. Goggles, eye protection. Lab coat. Tongs. Heat in water bath, no flames, reducing sugars, ethanol. Tie hair. Point tubes away from people. Fume cupboard. Dash. Cut on solid surface, cut away from body, avoid fingers. All healthy, suitable footwear, check for trip hazard. Benedict's reagent, crush, mix with water, heat. Ethanol emulsion test, shake. Anomalous result that does not fit the pattern trend. Investigation. IV, at least 3, suitable range. DV, CV, at least 3, type of plant. Method, prepare different IVs, collect samples, control CV, collect results, record, find change in XX, measure. Control, repeat and calculate mean results, collect at least 3 samples. Safety precautions. Conclusion, prediction.